All right, so now we are really going to jump into step 3 of cellular respiration. And I do kind of want to hit again on some of the things that I talked about last time and make some clarifications. So I did want to talk to you a little bit about bacteria and how this works in bacteria. So originally, I think I had told you that bacteria aren't going to do this because they don't have mitochondria. But I wanted to dig in a little bit more and do a little bit more research because they really do actually do this process. It's just done a little bit differently. So glycolysis, remember, where does glycolysis take place? In the cytoplasm, right? So hopefully you remember that in the cytoplasm, and really we've got the glucose is being broken down in the pyruvic acid or pyruvate, and then it gets shuffled off to the mitochondria. Well, like I said before, we know those bacteria don't have mitochondria, but they are going to have the enzymes that are going to help make step 1.5, which is that pyruvate to acetyl-CoA, and the Krebs cycle. They're going to have enzymes that are going to do the oxidation of those molecules, right? Pull off those electrons to break it down and produce these different products, right? And because they're doing oxidation, they're pulling off electrons, we have to have an electron acceptor. So the process is very similar, but because it's just in the cytoplasm, it's just not going to be as efficient as it is in eukaryotes. So it happens, all right, this process is going to happen in prokaryotes. It's just not going to be as efficient because it's just taking place in the cytoplasm and the enzymes aren't necessarily all organized in one place. So that's really kind of what I wanted to clarify on. I think that is something that's important that you need to note, that, we have these similar pathways again, which when we're seeing things that are similar, that's really pointing us to the idea of a common ancestor. So this is another place where we're seeing evidence for a common ancestor. All right, so step 3. This is for eukaryotes in the inner mitochondrial membrane. For prokaryotes, this electron transport chain is going to just be in the plasma membrane. All right, and we're going to have these different the electron transport proteins that are going to allow the electrons to pass through. So it's embedded in the inner mitochondrial membrane for eukaryotes, and we're going to go through this process, but really this process is a review of what you've already seen in photosynthesis. The biggest difference is we don't have photosystems exciting the electrons to move them along to the next place. So we just have these proteins here in the membrane. Okay, so where do we get these from? This is another thing that is different from photosynthesis. So these electrons are going to come from our checks. Remember we talked about how we had our check pile and a cash pile? Now this is where we're taking those checks to the bank. All right, so we have NADH and FADH2. Remember the NAD plus and the FAD, those were our electron acceptors, and they became NADH and FADH2. This is what we're going to pull off to get electrons. We're going to pull off hydrogens, and those hydrogens are going to split into protons and electrons. And they're going to do the same thing that they did in photosynthesis, right? So the electrons are going to go through this electron transport chain, and the protons, the energy from those electrons moving through the electron transport chain, are going to pump out the protons. And think about what happened to those protons in photosynthesis. Okay, so we'll keep going, okay? So those electrons, like I said, those electrons get pulled off and pass through the electron transport chain, are used as energy, but they need a place to go. We need a final electron acceptor, and that's going to be oxygen. that sometimes is called terminal. That's another word you might see instead of final, but oxygen is going to be the final electron acceptor, and it's going to combine with protons, the electrons, and those are going to combine to make water. All right, so this is where water comes in. So this is one product of the electron transport chain is going to be 2 molecules of H2O. All right, like I said before, that energy from those electrons passing through the electron transport chain are going to pump those protons into the inner membrane space. So now we have this electrochemical gradient. We have lots of hydrogens on one side compared to the other, and they're just going to naturally want to flow through. And they're going to diffuse through that molecule. Remember that enzyme ATP synthase? And that process of them diffusing through is called chemiosmosis. All right, chemiosmosis. So the protons passing through ATP synthase, and is going to power the enzyme to make ATP from ADP. Okay, this type of ATP synthesis, you need to know this terminology also, is called oxidative phosphorylation. Remember what Leo is, right? Leo the lion there. Loss of electrons is oxidation. So that makes sense, right? We are using energy from the electrons, from losing electrons, to phosphorylate that ADP. Okay, so if you think about some of this terminology, it really makes sense. So oxidative phosphorylation, Leo, right? Loss of electrons, oxidation. So like I said, I think remembering Leo the lion is really going to be beneficial to you because they are going to refer to reduction and oxidation, and you need to have an idea of what I'm talking about. All right, so this is electron transport chain here. So like I said before, looking into prokaryotes, comparing it with prokaryotes, same thing's really happening, but the biggest difference is, in this case, this is happening in the cell membrane instead of the mitochondria. All right, so overall, what's happening again with the electron transport chain? So it's very similar to what's going on in photosynthesis. The big difference is, we've got our checks where we're getting those electrons from to power this process is from a different molecule. So instead of water, like it was in photosynthesis, we have NADH and FADH2. That's our checks. So we're going to cache those in, and we will pull off the protons and the electrons flow through the electron transport chain. That energy is used to pump out the protons. The protons are used to power ATP synthase to make ATP. Okay, those electrons need somewhere to go, so they're going to combine with protons and oxygen to make water. And our payoff ends up being 28 ATP. So we get big money here. So hopefully you have an idea of how many we've made in each of these steps so far. That's something you probably should take into account. Think about all the total. All right, so that's something that you need to kind of keep track of. But this, like I said, this is where we're going to the bank. We're going to make some money here. All right, I'm going to go ahead and pause here.